Last but definitely not least, question 8, your lovely 9 marker, where your quality of English will actually be taken into consideration a little bit here. Key thing for that one, as with all the others, read it, structure it, and then you're writing. So before you've actually done the reading, don't let your brain fall into the pattern of, oh, I know stuff here, I'm going to write it. Read it first, structure it. I've seen a lot of little snippets of planning notes when I marked your paper, so thank you for that. It is starting to happen. It's great. So what does it give you? It gives you the idea of the purpose of the Human Genome Project as a general understanding genetic disorders, as a general what is going on. The original study gives you additional information to what the actual textbook stroke your syllabus has given you. The original study was a small number of anonymous donors from Europe. We do not know anymore. Okay. Then, we've got 100K projects being set up. These ones are now the specific with regards to getting rarer genetic diseases and cancers and analyzing and sequencing the human genome with regards to identifying those codes and putting up a basis into place as to how can we potentially treat those particular genetic conditions and cancers. Now, it gives you a map where in the world these rare genetic diseases and cancers seem to be important enough to spend that amount of money to get a 100K project going, or to get those 100K projects going. Ours has been funded by the NHS, but it's a question of where, first of all, is the money available, and the map will kind of speak for itself, where the money is available and where it isn't. But also, where is that a priority compared to other research? Now, other research is kind of given down there. Other research could be a genetic research genome of mosquito, or the actual plasmodium that is the actual parasite which is transmitted by the vector, the mosquito, and the malaria being responsible for millions of deaths. It doesn't say where. Map should kind of speak for itself a little bit. We have looked at maps as to where malaria is prevalent. Now, structure of your answer, it gives you a describe, and explain, and suggest. It gives you a 3-part structure. So if you're thinking your 9 marks could be split up into 3 marks for first, second, and third, that's not a bad way of going about this. So the first one, findings of the Human Genome Project, how could it improve further treatment, and why the findings are of limited use. So first of all, how could it improve the treatment of human disease? So literally knowing about how many genes do we even have, where are they, what does that help, and suggest findings of how they could be of limited use. That sentence up here, that should literally give you a bit of a hint as to what will be one of your marks available to limited use. For the first one, just look at the mark scheme. Again, let's look at it in the 3 parts that the question came like. It helps you identify, give diagnosis, predictions. Does it actually allow you to treat anything yet? But maybe not. But it may at least allow you to make genetic decisions as to the next generation with regards to who would be having to look out for what conditions right away as from birth or so on, or make other decisions. The sample size is way too small if you've got a few anonymous donors. If you know that they're all European in origin, we don't have anyone there who's Australian Aboriginal. We don't have anyone who's Polynesian, Southeast Asian islands, for example. We don't have anyone who's from a wider ethnic background in there. So that's not there. Also, because it's anonymous, we don't actually know what genetic conditions they have. We don't know what they potentially have died of, because they might have had genes that relate to genetic disorder or cancer. So that is for the first one. You don't have to have hit all of those to get the kind of 3-mark equivalent. I'll explain the marking in a little bit, because it's not straightforward, and I can do 3 bullet points here, gives you 3 marks. It's not that straightforward. From the second part, explain why the 100K projects will eventually provide more valuable information. So this is literally pros and cons again. Like with the first one, what is great about it and what should be done next, because it's not quite there yet. Why more projects are needed to be set up to improve the treatments on a worldwide basis. Worldwide basis. the map kind of gives you a little something. You're saying, actually, you know what, not all countries have been able to or have chosen to, whichever way around, to invest in a 100K project. Where will it provide more valuable information? So in order to treat all those various cancers and genetic conditions, if you know what the sequences are that cause a condition, then you can potentially come up with a treatment, or you can test for it and look at reproduction in the next generation. And gene counselling, genetic counselling to go with that. The mark scheme expresses it like so. People in the sample selected are based on their genetic conditions, which is actually a good thing, because like that, you're going to be able to identify a lot. The larger samples are there if it's 100,000 people. That is a larger sample, definitely, compared to the few individuals for the Human Genome Project. What does it include? It's still a bit vague as to what does it include. Have we got more of the ethnicities? Have we got male and females? The different ages is a bit of a wobbly one, because yes, you do get some change to the genetic structure as you go through life. So it's valid to have that here. The projects do not include any areas of Africa or South America, really. So is it representative of the whole human population? Not really. Not really. But possibly maybe there's a reason why some countries have not invested in that. And that gets us to the third part here, the suggest why some countries may have invested more money into sequencing the genomes of malaria and parasite. The malaria parasite. The actual Anopheles mosquitoes have been, the Anopheles gambiae have been sequenced as well in that sort of thinking. And the vector, here we go, that's the Anopheles gambia. Rather than the funding of 100K projects, think of different countries and where do most people die, and what do they die of? What conditions are actually causing the health service and the society as a whole most of a burden, whether that's financial or emotional, or just simply by losing people? And therefore, you may want to invest money differently if there's money around. This is again asking you to kind of suggest, is asking you to think out of the box a little bit and seeing planet Earth as a planet as a whole and societies as a whole. So malaria is still killing people at a drastic rate, and it's in some countries more than others that the malaria death rates are higher than the genetic rates. The improvement of drugs to treat malaria, the insecticide development, and the resistance against all that is still a work in process. It's still, and vaccines is all work in process, is not there, and not all of it is effective. So sequencing the genome of the Plasmodium and of the actual vector itself may give you a way forward in some sort of genetic way of treating it. I think we've had in the syllabus that they are considering putting the gene for an antibody into the Anopheles that would not be allowing the transport of the Plasmodium inside the vector anymore. I did say the marking is a bit complicated, because I have to look at your answer as a whole and decide, does it go into, yeah, one area has been covered fairly well, or two covered kind of okay, but relevant points are made, but not actually full reasoning. So showing some limited reasoning, some mistakes maybe, some omission, so significant bits of that mark scheme missed out. And then within that, I'll have to make a decision. Is quantitative English good enough? Is all of it there? Moving on, obviously, 46, there's a whole lot more coverage of two areas, and maybe part of a third one. They're kind of a step up, and some questions have been addressed, not that many omissions, so loads of relevant points missed. And for the top marks, what I'm looking for is a proper logical arguing of most of what was in the mark scheme up there. Some, because of this, therefore that's going to happen. That's what's meant by sequential reasoning, because in some countries the death rate from malaria will be higher than the genetics and cancer death rate. Therefore, investment in a 100K genome project may not be most valuable in that country. So, yeah, vocabulary, appropriate reason, just a reasoning how you're doing that. That was component 2.